Hello, IB Bio students. This is Ms. Sheely with your next edition of Notes. Today we're going to talk about proteins, which is section 2.4 of your 2016 IB edition book. All right, so proteins are polypeptides. They are polymers. So the monomer of a protein is called an amino acid group. When we look at the generalized structure of an amino acid group, you have on the right-hand side in the red, you have an amine group, which is a nitrogen and two hydrogens. You have a central carbon with one hydrogen off the bottom, and at the top is an R group. This is interchangeable. There is no element R on the periodic table, but there are many different amino acids, and so putting this R here indicates that this is an interchangeable unit. And then here we have our carboxyl group with our double bonded carbon oxygen, and then our hydroxyl. So as I just said, this R group is interchangeable. If you look down here, each of these amino acids has the amine, central carbon, and carboxyl group on it. Here's your amine, your central carbon, and your carboxyl. Doesn't matter how it's written. You notice that each of these carboxyls is written a little bit differently. But if you look in here, your R group is completely different for each. The cysteine has carbon, sulfur, hydrogen. Your glycine just has a hydrogen, and then your asparaginine has carbon, and then carbon bonded to oxygen and nitrogen. So we still do condensation and hydrolysis, just like we did with carbohydrates and fatty acids. So you take your monomers, you're going to do condensation, release water. You're going to use an enzyme, depending on which what we're making, which proteins we're making, is what the enzyme is. So this is generic synthase. Take your dimer, do condensation, release the water, and then you can also turn it into a polymer. And then, of course, looking at the reverse, we go from our polymer, put water in through hydrolysis, put water in through hydrolysis using an enzyme. We can go from our polymer to our dimer, and then doing the same, putting water in, using an enzyme, go from our dimer to our monomer. So this is all the same process. Now we're just focusing on our monomers being amino acids and our polymer being proteins. So how exactly do we do this? What does it look like when we take these amino acids and make proteins? So here are your two amino acids, and then there is your dipeptide. So when we do our condensation, we're going to take the hydroxyl group from the carboxyl, and take one of the hydrogens from the amine group, put those together. Those are going to become water. And then a bond will form between. This bond is a peptide bond. And then here's our water product. Here's a little animation. There you go. Seeing it in motion. All right, so proteins, we get this amino acid chain, and we call it a polypeptide chain. That's our protein. But to actually get the fully functional protein, it needs to be a folded three-dimensional structure of the polypeptide chain. So we start with the first level of protein structure, which is a primary chain, and that is just the amino acid sequence, and the amino acids are held together by peptide bonds. So that's called primary structure. We go from primary to secondary, and then we're going to have a repeating local structure. This could be a helix or a spiral. This is called an alpha helix, or it could be a pleat, and this is called a beta pleated sheet. The beta pleated sheet and the alpha helix are held together by hydrogen bonds. You can see these little hydrogen bonds here. And then there are hydrogens as well, hydrogen bonds as well, holding these sheets together. We go from the secondary, the beta pleated sheet or the alpha helix, to a tertiary. So this is where our protein becomes folded, and this has to do with the R group interactions and forming a hydrophobic core of the protein and having all those hydrophilic amino acids on the outside, and so that it can function, the protein can function in our cell. And then sometimes we go from a tertiary to a quaternary, and this is when we have a protein complex. This is when we have two or more subunits of tertiary structure put together. So here you can see hemoglobin. Hemoglobin actually has 4 subunits, which carry the oxygen molecules in the red blood cells. All right, so let's look at primary structure a little bit more. So primary structure is, like I said, the polypeptide chain. So it's just your amino acid sequence and held together by peptide bonds. When you're looking at a primary structure, you should always be able to see the backbone. All right, so when we're looking at the backbone, you should be able to see the nitrogen, carbon, carbon. So in this case, we have an NCC, NCC, NCC. So in this case, we have 1, 2, 3 fully functional amino acids. There is a 4th and a 5th amino acid that are attached, but you don't see the entire thing. So let's think about it. You have 20 different amino acids, and proteins could be any length. You can have proteins from just 5 amino acids all the way up to the average length of most proteins are about 300 amino acids or more. And those amino acids can be in any order, and they all have different properties because of the order. So there's an infinite possibility of polypeptides. And that's just the polypeptide chain, not even focusing on the folding. So let's look at this animation of primary structure. So again, you see the NCC peptide bond, NCC peptide bond, NCC. And then all the side chains are always on the same side of the molecule. All right, so polypeptides are chains of amino acids joined together by peptide bonds. Cannot stress that enough. There are 20 different amino acids, like we just said. These can be combined in any order. Each amino acid has unique properties. Some are hydrophilic, some are hydrophobic, some are positively charged, some are negatively charged, and there is one that contains sulfur. So the properties of these amino acids are going to determine how the polypeptide folds up into a protein in that secondary and tertiary structure. So secondary structure is when we have those repeating patterns, the beta pleated sheet or the alpha helix. So these are both held together by hydrogen bonds. So here is your alpha helix held together by the hydrogen bonds. You can see that in the spiral here, and then here as well. And then the beta pleated sheets are these silver areas, the pleats on this protein. And you can see this example here, this one protein, one polypeptide chain has 2 helices and 2 beta pleated sheets. So there's not that the whole protein needs to have one or the other. All right, tertiary structure, that's when we get into the 3-dimensional folded structure. So this is making that hydrophobic core and having to do with the interactions of the proteins. So this 3-dimensional structure is going to give our proteins our functional properties of the protein. It gives the specificity to the enzymes. So there are 4 things that we're kind of looking at or are happening for the protein to fold into its tertiary structure. You have the hydrophilic amino acids start to orient towards the outside, and the hydrophobic amino acids start to be oriented to be protected in the core of the protein. Then you have the oppositely charged ions attracting the positive and negatives, forming salt bridges. And then finally you have the sulfurs forming disulfide bridges. And then finally we can have quaternary structure, which is when we have multiple subunits, so just many pieces together. So we already talked about hemoglobin having the 4 different subunits. Ion channels typically have more than 1 subunit. So here we have 2 subunits, an A and a B. And then even your DNA polymerase replicating the DNA has many units to it as well. So let's take a second to look at this, starting with primary structure. Again, magnifying, seeing those peptide bonds. Primary going into secondary, you have beta-pleated sheet and alpha helices. Now folding into tertiary, and then two coming together to form quaternary. All right, now that we've talked about how proteins are formed, let's talk about the two major categories of proteins. You have fibrous proteins and you have globular proteins. Most of the proteins that we will be talking about throughout the year or the 2 years are going to be globular proteins, but there are many fibrous proteins that are also very, very helpful. The first big difference is that fibrous proteins are insoluble in water, whereas globular proteins can be soluble. There are some that are insoluble as well, but most of them are soluble. Fibrous proteins are typically structural proteins. They help with support and strength of the organism or the cell, whereas globular proteins are functional. They could be enzymes or even antibodies. Examples of fibrous proteins: keratin in our hair and nails is a fibrous protein. Our elastin in our skin is a fibrous protein. And then collagen, which is a tissue strengthening protein, is fibrous protein. Examples of globular proteins: amylase, which is used to digest starch. And notice, look, it ends in ASE, so it is a enzyme. Insulin is important for blood sugar regulation. Hemoglobin, this is the British spelling of hemoglobin, that's why you have that A there, carrying the oxygen around in our bodies. You have immunoglobulins, which are your antibodies, and then you have transport proteins, for example, your sodium-potassium pump, and we'll come back to that later in the year. So how do we use proteins? Proteins can be structural or part of this plasma membrane, and they can also perform many other functions in the organism. So we just talked about some, but there are hormones such as insulin, there are enzymes such as catalase, which we'll look at, there's gas transport, which is hemoglobin, and then immunoglobulins, antibodies, and in charge of infection fighting. The polar and non-polar amino acids are really important in the folding, and in particular, they're key in forming the membrane in channels. You have a polar section, it's going to be the opening of the channel, and that is to allow ions through. Charged or polar molecules can't normally go through the hydrophobic layers, which we'll get into more when we talk about cell structure. So the nonpolar sections bond with the hydrophobic tails to keep the protein in place. Polar amino acids are positioned on the external surfaces, and they also line the protein channels, and this helps for facilitated diffusion. Let's look at a channel protein. So we're going to start with the non-gated channel protein. So you can see here we're going to have the hydrophilic pieces of the amino acids here on the inside. That's going to allow the potassium or whatever is going through, as well as any cytosol. And then let's look at the gated. So in this case, you're going to have hydrophilic also here on this top outer flap, this gate, but then also the inside structure will have that protein structure. All right, and that's it for proteins. Super short today, so see you in class.